Thank you, everyone, for the opportunity to be presenting my 10-minute talk. So here are my disclosures. So as I said in my pitch, around 80% of the children diagnosed with cancer live in low and middle income countries, where the access for precise diagnostic tools, especially when we talk about molecular diagnostics, is often out of reach. So what if we could have something that is fast, simple, and affordable, and be able to diagnose a wide range of tumors anywhere in the world? So my project is part of this DARGAN network, which is Diagnostic Applications in Resource-Limited Settings Using Global Advances in Nanopore Technologies. This collaboration is being led by St. Jude and the University of North Carolina, and it involves a lot of countries around the world. And our institution in Brazil, Barretos Cancer Hospital, is the one representing our country. So the collaboration already published two significant papers using this approach for classifying pediatric leukemias and pediatric solid tumors. So here we wanted to expand this approach and include a new tumor type, which is the pediatric germ cell tumors, and expand this cohort. So talking a little bit about pediatric germ cell tumors, they are a rare type of cancer, and they account for around 3% of all diagnosed tumors in children. They usually arise from primordial germ cells, and they can differentiate into several histological subtypes, which are very important for tumor clinical management and prognosis. They appear along the midline of the body and the gonads, and their diagnosis is usually based on morphological evaluation by a pathologist. And there are few serum markers that could be used to help the management, like alpha-fetoprotein and beta-HCG and LDH, but they are not specific at all. So for our population, we got 47 cases of pediatric germ cell tumors, including 27 only FFPE samples, 11 only cryopreserved, and from 9 cases, we got both sample types. So we had 9 paired samples. The study was approved by the ethical committees from all participant institutions, and our inclusion criteria was the age between 0 and 18, and the availability of the tumor tissue, either frozen or FFPE samples at our biobank or the pathology department. For exclusion criteria, some of the samples that we extracted RNA had low quality or low amount, and some of the samples were evaluated by a pathologist and had less than 60% of tumor area or more than 20% of necrosis, so those samples, unfortunately, were excluded. So here's my workflow, and I'm going to walk you through a little bit. We first extracted the RNA using semi-automated Kia Symphony and the manual recoverall kit for the FFPE samples and quantify it using Kilbit. So then we synthesized the cDNA using the PCR cDNA barcoding kit from Oxford Nanopore Technologies and amplified the samples. And this kit allowed us to sequence up to 12 samples per run. And we used the R9 flow cells on the MinION device. It's important to say that all our cryopreserved samples were sequenced separately from our FFPE samples. For analysis, we used Dorado-based color in super high accuracy mode, and we align our reads with Minimap2 to the ensemble transcriptome and built the gene expression matrix. For the classification approach that we've been using, we use the machine learned model that was developed by UNC for the leukemias and other solids. that is composed of a partial least squares and support vector machine, and we use the leave one out cross-validation approach. Here we also implemented a calibrated classifier CV from scikit-learn to improve and make the probabilities reflect the reality of the classifications and the true results. So moving to the results, most of our patients were female with primary localization in the ovaries. Most of them were classified as a low risk with no metastasis at diagnosis and no relapses. As for sequencing results, we had a pretty shallow sequencing. Our N50 mean was 204 base pairs for our FFPE samples and 337 base pairs for the cryopreserved. And we had around 0.7 million reads per sample. So for the training and testing of our classifier, we used 52 samples. 4 of them were mixed cases, so we labeled them according to their major subtype composition, as long as it had more than 70% of that subtype. And we also grouped our teratomas together because we had both mature teratomas and immature teratomas, but they were grouped together here. And we also filtered out the genes with 0 counts in at least 10% of the samples for each class. So we retained around 6,200 genes. So here is our first try. We got a good overall accuracy, but we got consistently low prediction probabilities. So our overall accuracy was 90.3%. So now we wanted to make the prediction probabilities reflect better the model's performance. So that's why we decided to implement the calibration step. So we, improving this, we got accuracy of 93.7% above high confidence cutoff, which is 0.8. So that was really good. We were really happy with that result. And in terms of performance, our model had high F1 scores for most subtypes, except for our embryonal carcinomas. But that could be explained by the low sample size of that subtype because we only had 4 samples. We also had AUC values closer to 1, meaning a high capacity to distinguish between the classes. And we also wanted to check a little bit about the differences between the FFPE and cryopreserved samples. So we used those 9 paired samples to perform a blend-Altman analysis, and that showed more than 95% of agreement between the gene expression profile of the paired samples, meaning that we can and we are able to use them together in the classifier. And in that sense, I also trained the model with only my FFPE samples and tested on the cryopreserved ones, achieving 100% accuracy. As for next steps, we added the germ cell tumors to the large solid tumor cohort that was already published, and that was really good too. And we started external an external validation with 5 samples that we obtained from Dartmouth College, thanks to Dr. Chanche and Dr. George Zanazzi. So using our classifier and testing on those samples, we got, out of the 5, 3 were correctly classified, and one germinoma was classified as an embryonal carcinoma with a low confidence, which can be explained by the similarity of gene expression profiles between those 2 subtypes. And the other misclassified sample was a teratoma, and it turned out to be an adult case, and all of our other samples were pediatrics. We also tested on the larger solids classifier and got a very good accuracy and high prediction probabilities. We are now looking forward to validating those results with additional samples with samples from institutions that are affiliated with the AMART Alliance, which is a network of innovation and collaboration in research and diagnostics for cancer in children. And with that, I'd like to thank everyone that participated and helped in this project, and also our financial support. Thank you.